I think Gaia will start first.
Okay.
That's okay.
Yeah, I don't mind. I really don't mind. If you, Kirsty, if you need to run off or if you have like clinical commitments, I'm also fine with swapping. It's entirely up to you.
No, it's good. I've cleared this.
Okay. I want to hear you speak, so I don't mind whichever way.
Exactly.
Exactly. Yeah.
So I will then mute.
Okay, now we are also on YouTube live. Okay. Okay, so I think we can start whenever you feel, Tara and Evan.
Okay, let's give it a minute. It seems like some people are still coming in. Okay, I think I am happy to begin. Yeah. So welcome everybody to this UCT Oxford Cortex Club Neurodevelopmental Mini Symposium. Thank you all for coming. We have a good audience at the moment. We have 54 people online, and I'm sure some will be joining us as we go along. This event has actually been in the planning for many months, and we are delighted that it's finally come to fruition today. My name is Aaron, and I am the current chair of the UCT Cortex Club. So the UCT Cortex Club was actually founded as a branch of the Oxford Cortex Club, and it was established several years ago. Traditionally, UCT have had a very strong neuroscience connection with Oxford, with the likes of Dr. Joe Raimondo and Dr. Richard Berman, who's also on this chat now, I see, being some of the most recent scientists who have gone over to Oxford. And we have one of our very own committee members, Andrew Tucker, who is set to go over on a Rhodes Scholarship within the next year. In Cape Town, we've also recently hosted Professor Gabriela De Luca and Professor Daniel Anthony, while Prof. Tim Vogel's co-organizes and attends the computational neuroscience Imbizo gathering every year in Cape Town. So clearly we have quite this rich neuroscience connection between us, and we hope this event and future events will help strengthen the synapse between our two good societies. So first, I would like to just set some Zoom ground rules. Firstly, please can you keep your video and audio off while the speakers are talking, just to allow them to talk without any background disruption and background noise. We kindly ask for you to please keep your questions to the end of the talk, where we'll have a brief question and answer session after each speaker finishes speaking. Should you miss the opportunity to ask a question or only think of your question a bit later, feel free to ask it in the virtual pub chat later this afternoon after the talk. So I think now I'd like to hand over to Tara, who will introduce herself, and Prof. Gaia Scerif.
Thanks, Aaron. Hi, everyone. My name is Tara Diviney, and I'm the current Freshers Officer and the co-president-elect of the Oxford Cortex Club. For those of you who don't know, the Oxford Cortex Club was established in 2009, and we were delighted to recently celebrate our 11th birthday virtually with some of the members of the Cape Town Cortex Club. And for today's joint mini symposium between the two societies, we are very excited to have two excellent researchers, Professor Scerif and Professor Donald, join us. On behalf of the committee of the Oxford Cortex Club, I'd like to thank Aaron and all of the committee of the UCT Cortex Club for reaching out to us to make this collaboration happen. And I'd also like to thank Dr. Richard Berman, a former president of the UCT Cortex Club, who's, as Aaron mentioned, is now doing his DPhil in Oxford at Colin Ackerman's lab. Although we hadn't originally planned this event to be completely online, we're still really glad that we got to make it happen today, and we hope that it will be the start of many collaborative events between the two clubs in the future. Just to add to Aaron's housekeeping rules, just to be aware that we are streaming this event live on YouTube. So if you would like to ask a question at the end of the talk, please be aware that your video will be on YouTube. And if you would like to keep your videos on during the talk, that's okay, but please keep your microphones on mute just to avoid any disruptions for the speaker. So without further ado, it is my pleasure to welcome Professor Gaia Scerif, Professor of Developmental Cognitive Neuroscience from the Department of Experimental Psychology at the University of Oxford. Professor Scerif did her undergraduate degree in psychology at the University of St Andrews and completed her PhD at the Institute of Child Health at University College London under the supervision of Professors Annette Kamiloff-Smith and John Driver. She went on to do a visiting fellowship at Cornell University and subsequently took a lectureship position at Nottingham University. And in 2006, she joined the University of Oxford. Professor Scerif leads the Attention, Brain and Cognitive Development Group. And so her research is focused on the development of attentional control and its interplay with memory and learning and mechanisms underlying attentional difficulties. She's also passionate about science communication and public engagement and has been the recipient of many awards, including the British Academy Rising Star Engagement Award, the Margaret Donaldson Prize for Outstanding Contribution to Developmental Psychology, and the James S. McDonald Foundation for Understanding Human Cognition Award. And we are so delighted that she has taken the time to join us this afternoon to give a talk on her research surrounding the use of developmental cognitive neuroscience tools to investigate mechanisms of atypical cognitive control. So I'd like to now invite Professor Scerif to share her screen and we can kick off.
Thank you so much. While I get this working, thank you very much to both committees and to the Cortex Club in general for the invitation. I'm so excited. Oh, I'm so excited about today, but actually I need to, as I was promising Tara, find the beginning of my presentation. I warned them that this was going to be interesting. There we go. So I'm so excited to be here. For many reasons. Well, first of all, the silver lining of lockdown has been that actually we've been interacting with people who would be much harder to meet in person had we not moved to so many electronic meetings. And so I'm really excited about the international aspect of this workshop. I'm also really, in many ways, drawn to reflect on my own title to begin with. I've changed it slightly because as I was preparing my talk, I realized that the use of the term atypical in a way indexes what's still very common, I think, at least in my area of developmental cognitive neuroscience, which is that we reflect and we study typicality amongst high functioning, high income, Western educated, rich and democratic participants. Weird. And not far enough study neurodiversity, both in terms of cross-cultural perspectives, international perspectives, and also neurodiversity within our own cultures and nations. So In no way is my seminar going to debunk that perspective, but I wanted to draw neurodiversity throughout. And so, as Tara explained, I am very much an attention geek. I'm really interested in attention. Why is that? There are many models of the cognitive neuroscience of attention and control in adults. I have my favorites, but I'm not going to reveal my cards. What I want to nonetheless start with today are what I think are some really interesting similarities across all of these models. One, they're characterized by varieties, differentiation in types of cognitive control mechanisms and attention mechanisms. Unfortunately, that's a different talk to the one I'm going to give today. I'm not going to be talking about varieties of attention and cognitive control in the interest of time, but I will focus on the second element of commonality across all of these models, the idea that attention and cognitive control act as a gateway to action, memory, learning, and therefore influence what we do, what we learn, and what we remember. However, increasingly, even in models of adult cognitive control and attention, it's become very clear that this is not a unidirectional stream. What we know, what we learn, what we've acted upon then influences how we attend as adults. And that idea has very much influenced the thinking with a colleague, Dima Amto, who's based in the States, but who shared my office when I was a PhD student, so many, many years ago. We found ourselves reflecting on the idea that if what we learn and what we remember influences attention, and in turn attention influences what we remember and learn, even in short online bursts as adults, what happens when we start to include time development into this thinking? As children learn and remember, how do they then attend better? And in turn, how does their attention influence what they learn and remember over time as they become older children, adolescents, and young adults, and elderly adults as well, whom we tend not to study, but we should as developmentalists. So my take-home message for the rest of today is going to be that while attention and cognitive control influence what we see in the environment, how we process it, in turn, what is in the environment influences the developing system. And so this dynamic, this interplay is going to be a common framework throughout all the levels that I'll try and talk about today. I'm very lucky to work with very many clever early career researchers, all the researchers, mentors who like to use very many methods to study these questions. We use EEG, eye tracking, and various imaging techniques. I don't do this all. I rely on some great collaborators, and we've asked questions about neurotypical processes as well as neurodiverse processes. Now, I think this interplay is necessary. In order to understand neurodiversity, we need to understand what's so-called typicality and vice versa. But for today, I'm going to focus on neurodiversity in particular. And I'll start off with actually delving into work that I did a long time ago with collaborators in Nottingham, so even before I moved to Oxford. But that has continued, in fact, to lead to really interesting insights. And the focus will be on ADHD in the first part of the talk, with some highlights on how extrinsic motivations, how rewards in the environment can and do change how kids with ADHD control their attention. And the second part of the talk is going to instead focus on children who are at very high genetic risk for having attention differences. And I'll highlight their variable outcomes across those children and close with some more recent work, which has entirely been serendipitous, but has brought me to think about different kinds of risk, environmental risk, particularly socioeconomic risk. And that's why I'm so excited, in fact, to hear Professor Donald speaking today, given that she's been at the forefront of leading work on understanding the impact of poverty and socioeconomic risk on brain development. So mine will not be, will just be a very short introduction and draw inspiration from her talk. So let's start off with ADHD. Now all this work was, again, a very big collaborative project. And here I just present the main players. I'll thank them all as we go along. Just as a brief outline for attendees who are not clinicians, ADHD is diagnosed behaviorally dependent on the behavioral symptoms that children present. I won't go into the details, the controversy, the consensus about ADHD diagnosis, other than highlighting that in attention and hyperactivity, both characterize ADHD. Some children are primarily influenced by inattention difficulties, some by hyperactivity difficulties, but in fact, those symptoms change over developmental time, and the majority of children with ADHD have a combination of both. Now, as neuroscientists and as cognitive scientists, behavioral diagnosis are only a starting point. What underpins, what underlies those difficulties in inattention and hyperactivity? Now, there's a huge literature that in the context of the neuroscience of ADHD, I can't even begin to summarize right now, but I want to draw attention to one particular characteristic of the literature so far. Work on structural differences, brain structural differences, functional differences, pinpoint atypicalities in the frontostriatal circuitry in brains of kids with ADHD compared to children without ADHD, both structural and functional differences, mapped with different kinds of imaging methodologies. And those have also drawn the attention, and another aspect that has drawn the attention of researchers in ADHD on the frontostriatal circuitry has been the response to medication, so the psychopharmacology of ADHD, with the majority of cases being sensitive to methylphenidate as a treatment of choice. Methylphenidate is the dopamine uptake reuptake inhibitor, and so dopaminergic circuits and frontostriatal circuits have been very heavily involved and studied by researchers who've been interested in understanding ADHD. And similarly, at an even lower level, a focus has been on candidate genes that have been particularly associated with dopamine, even though, and I highlight it here in case there are interested attendees who want to draw this for discussion, the evidence from genome-wide studies has been a little bit surprising and, in fact, interestingly, not converging on dopamine-only markers of risk in ADHD. But at the time when I first started working with ADHD, the emphasis and the focus was on core atypicalities in frontostriatal circuits in kids with ADHD compared to kids without ADHD. And at the cognitive level, that focus on the frontostriatal circuitry was also paired with a big interest in inhibitory control deficits, difficulties in controlling one's attention and responses and behavior. But even at the time when this core inhibitory control account was put forward for ADHD, there were warning voices. The suggestion that all children with ADHD present difficulties in marker tasks, in classic tasks of inhibitory control, and also an interesting reflection on differences in how kids with ADHD may process rewards and motivations that are provided extrinsically, which is what motivated us to start this project, focused on how extrinsic rewards might modulate inhibitory control difficulties in ADHD. So, are inhibitory control difficulties in ADHD, in fact, impervious to change in motivation? That was a question that we came to as part of the study. What are the neurocorrelates of these effects of motivation on ADHD brains? And are there additional risk markers that might explain differences between kids with ADHD rather than between kids with ADHD and not neurotypical controls? So given that we in the clinic and as cognitive scientists experienced pretty wide individual differences across children with ADHD, what could account for a child with ADHD having more difficulties than another child with ADHD? And so we embarked in this project, which is entitled MIDAS, Motivation, Inhibition and Development in ADHD. It focused on multiple levels of description, from cognitive measures all the way to structural measures and functional measures using MRI, fMRI, and ERPs, as well as a treatment aspect, the pharmacological manipulation on and off methylphenidate, and an investigation of genetic markers. So this was a study that involved very many collaborators, many of whom were clinicians and were involved in the assessment of the children who took part in the study. And ultimately then, we included children who had a full diagnosis of ADHD with the exclusion of other comorbid difficulties, and children who did not have a diagnosis with ADHD, who are labeled as controls from here on. These children engaged in very many games and tasks. Children with ADHD were on and off their medication. They took part in an ERP study and in an fMRI study, and controls equally took part in 2 sessions to have multiple measurements. from match controls in the study. So it was pretty big involvement on the part of parents and children, and we're very grateful for their contribution. Let's start off with the structural findings of that study. And here is Martin, who actually led that element of the study and got very many kids to be quite happy and not anxious lying in a scanner. That wasn't at the time all that much of a given, especially in kids with ADHD. And then we carried out a number of very classic and then slightly fancier in structural imaging analysis. And if you're interested in any of the details, please ask me later. I want to give you some take-home messages of what the structural differences told us, because they converge with what was already known about ADHD structural differences. We found that there were global differences. Children with ADHD had smaller whole brain volumes and lower gray matter volumes in all lobes that we studied, but there were also some localized differences over and above those, and they were in the classical cognitive control areas, like in fact the inferior frontal gyrus that was thinner in terms of cortical thickness in ADHD compared to controls. And the findings were very rich, and in fact, I urge you to contact Martin, who was really excited and interested in replicating previous findings in this literature. Nothing new. The brains of kids with ADHD are different to the brains of kids without ADHD. What we were, however, very interested in studying next was the extent to which, at a functional level, we could leverage motivation to understand the inhibitory control difficulties that children with ADHD encounter and hopefully change them, improve them. So we did this we were joking as a team because we thought at the end of this project, if we can't get a job in academia, we'll go into gaming, because part of the study involved designing a pretty cool alien game. These, I don't know if you recognize the characters, but they are actually from a cartoon that was quite popular amongst adolescents at the time, Futurama. I don't know if you know this, but the game involved responding to particular aliens, the green aliens here, but being very careful and not responding to the other funny aliens. And there was a narrative that surrounded this idea. Ultimately, this was a classic go, no-go task. There was some go stimuli and some no-go stimuli. But what we manipulated was how difficult this was, because we changed the temporal window within which people had to make a response and made it really difficult for people who were good at inhibiting, because we gave them increasingly less time to inhibit, and allowed children who instead had more inhibitory difficulties to have a bit more time to wait and really stop when a no-go stimulus appeared. So that at the end of this process, behavioral performance was equated among children with ADHD and children without ADHD. We thought this was really important, because part and parcel of being really bad at inhibiting is that you very often encounter failure. It's a really difficult thing, and that in turn might have an impact on functional involvement of brain areas. So we wanted equivalent performance across the two groups. But we manipulated the motivational conditions under which children played this video game. Under a neutral condition, it didn't make much of a difference. You were correct or incorrect on inhibitory trials or no-go trials. Under the reward condition, you were rewarded in a point kind of scenario for doing well on no-go trials. Instead, in the response cost condition, you received the penalty for incorrectly going when you were not meant to do. So this is a go no-go task with motivational incentives and a tracking algorithm to result in equivalent performance across groups. What did we find at the level of fMRI, functional magnetic resonance imaging? Again, the results are pretty complex. So I'll just give you what I thought were the highlights of that study. We were really interested in not only focusing on the frontostriatal circuitry, because already in the early 2010, there was a lot of interest in studying the ADHD brain more broadly in terms of networks. I know now, if you're a neuroscientist doing fMRI work, you only look at networks and not regions of interest. But at the time, like a few years ago, this was new. And we hadn't approached the idea of functional networks in the context of ADHD brains as much. Now, much more work has been done in this area. Elizabeth, who led the study and the analysis in particular for the functional imaging, was interested in all sorts of networks, but one particular network that was of interest at the time was a default mode network. This is a network that's quite broad in distribution. It involves the parietal cortex, the precuneus, stereocingulate cortex, and medial frontal cortex. But very interestingly, seems to be engaged in mind wandering, and it requires the activation when you're engaged in an active task. And so we found this was something that had been reported in the literature before, and not just in the context of ADHD, that when engaging in an active task, this network, the DMN, needs to be deactivated to do so efficiently. There's a lot of work on this, some controversies. We can get back to those if you want in question time. But what did we find for our children with ADHD? Well, we found that they responded interestingly differently to controls, to a motivation condition, with an interesting caveat that is associated with medication. So let me first say that controls show the activation of this DMN network, both when they're motivated and when they're not, via rewards. So control children were quite good at shutting down this mind-wandering network, regardless of whether there were high extrinsic rewards or not. Children with ADHD deactivated the DMN less well than kids without ADHD. So perhaps if you take this as a really simplistic proxy of mind wandering, maybe they weren't as good at deactivating the DMN when required. However, this deactivation was influenced by motivation. It was also interestingly influenced by medication, and medication and motivation had similar effects. So I'm going to use my pointer here. I'm just illustrating this activity differences at one particular point within the network, but actually the findings are across the network. It's easier to look at a graph like this than to look at a full network. You see controls, whether they were on the low or high inhibitory incentives, DMN deactivation is similar. They deactivated. Kids with ADHD who are on meds also deactivate the DMN quite efficiently, whether they are engaged in the low inhibitory incentive condition or in a high inhibitory incentive condition. But when kids with ADHD are off meds, the same children, high incentives are required for that network to be deactivated. That to us was very interesting, in fact, super interesting, because it showed that both medication and motivation had an effect on DMN deactivation. So it's not the case that kids with ADHD couldn't deactivate that network. They could if they're under medicated conditions, or if they were highly motivated to do so through extrinsic environmental rewards that were present and had an effect on them, but not as big an effect on controls. So those were really interesting findings in fMRI. Interestingly, they converged, in fact, in EEG, event-related potentials. Again, just in the interest of time, I won't spell these findings out in full, but the highlights when we collected EEG data in a parallel session, so these children went into the scanner, and then they also did a really long session of EEG. They were amazing. And we collected EEG and then event-related potentials of their performance on the task under a neutral condition, and then a reward condition, and a response cost condition. What you see in classic components that are associated with processing stimuli in a go-no-go task, the frontocentral N2 and the P3, is that children with ADHD who are off meds have lower amplitudes of those two components compared to controls. So the dark lines are controls here, and the dotted lines are children with ADHD off meds. But children on meds, and increasingly so when under reward conditions, ADHD in the context of ADHD show a higher amplitude that is in fact closer to the normal controls, so controls without ADHD. So altogether then, both medication and motivation, even in the context of a different imaging modality, EEG, show some influences on brain functioning in ADHD. Now, just to close off this first big part of the talk, we continue to be very intrigued in not only looking at how children with ADHD differed from children without ADHD and the effects of motivation, but also we were interested in the diversity across children with ADHD, because again, clinically, and in the lab when they came in, we could observe really interesting differences across children with the same diagnosis. And so it started to investigate in an exploratory fashion whether at least some of those differences may be associated with higher genetic risk characteristics. So this is very preliminary work. However, I wanted to acknowledge the work of Lena, who really was very interested in using our data as exploratory data and divided up the children with ADHD, but also controls, in children who carried risk alleles of various candidate dopaminergic genes. The RD4 is one of them, and there's a pretty extensive literature associating the 7R allele with higher risk. It's a mixed literature. We didn't know what we would find, but we saw some indication of structural differences between children with ADHD. And again, I'll point here in blue, differences between children without the risk allele and children with the risk allele in particular markers of structure. And we've investigated, again, in exploratory fashion, those same differences within our sample of kids with ADHD in other imaging modalities, fMRI, and also in the ERP data. Of course, these data were very preliminary, and we would really love to hear more from others interested in replicating them or using or combining samples to look at individual differences within samples of kids with ADHD. But what I've hopefully given you a flavor for with this data is convergence with previous data. Definitely, kids with ADHD are characterized by attention weaknesses, and those are reflected also in multiple structural and functional differences in their brains compared to neurotypically developing children. However, extrinsic motivators, rewards, can make a difference. So the environment that surrounds children with ADHD also has an impact on their brain functioning. And there are also differences between children with ADHD that need accounting for. And again, a lot of work in the last 10 years on ADHD has focused on understanding within group differences, presentation of children with the same diagnosis, but their differentiation. Of course, though, and here is a developmental symposium, ADHD is a developmental condition. However, there we only took one time point, one snapshot of difficulties and performance changes with motivation. We didn't study the children longitudinally. How best can we study changes over time? This leads me on to the second part of the talk, which is the focus on genetic risk, and studying genetic risk over time. And hopefully the significance and the relevance to attention difficulties like the ones with ADHD will come clear, and become clear quite quickly. Now, at about the same time when I was doing work with ADHD with colleagues in Nottingham, there was a lot of excitement because the Human Genome Project had concluded, because there was a lot of information on the functional role of individual genes in the human genome and the association with pretty severe difficulties that characterize individuals with big genetic hits. I'm going to refer to these groups as genetic syndromes, just as a shorthand for syndromes with identified genetic etiologies. They provided a lot of excitement because they provided the opportunity to study at multiple levels how a genetic difference might be associated with brain function differences, brain structure differences, brain function differences, and cognitive and behavioral differences. And now I'll leave this slide up for a little bit, but the one genetic syndrome I want to illustrate in the second part of the talk is Fragile X syndrome. This is because it's, genetically speaking, quite simple. It's monogenic in origin, associated with the silencing of a single gene on the X chromosome. So if you're a man or a boy, you only have one X chromosome. If the FMR1 gene is silenced, as it is in the context of Fragile X, that results in very limited or null expression of the protein that normally is associated with it. And that has very well studied now effects on dendritic spine morphology and many other structural and functional aspects that have been studied in animal models of the Fragile X, as well as functional and structural differences that have been studied in live human brains. And so it presents itself as an intriguing model in which to study the links and the relationships between genes to brains to cognition, and the other way around also. Interestingly, because the diagnosis for Fragile X syndrome can come very early in life, increasingly it's been studied as a model of developmental risk to change over time with longitudinal data. And that is in fact what I got very excited about as part of my PhD, and I've continued doing for many, many years now. So the genetic story, as I said, is quite simple. But what becomes really interesting when studying syndromes like Fragile X is that it is a very complex syndrome at the level of the cognitive and behavioral phenotype. It's associated with very high risk for ADHD symptoms. So up to 70% boys with Fragile X fulfill a diagnosis for ADHD. So going back to the first part of the talk, they're having attention and hyperactivity. That is very often in the context of intellectual disability. So we can return to this point later. Is ADHD in Fragile X like ADHD outside of the Fragile X realm? We'll come back to that point later if you want. Interestingly, Fragile X is also associated with a higher incidence of autism spectrum symptoms, so ASD. The diagnosis, again, is variable, but nonetheless higher in likelihood than it is in the neurotypical population. And this is all paired with a diagnosis that can be very early, earlier than the typical diagnosis of ADHD or autism spectrum might occur. I like to always illustrate this point with reference to a family who very kindly gave me this photograph, and I came into contact with as part of one of my first studies. These are two brothers who both have the full mutation for Fragile X. The eldest boy in the picture was 2.5 when he received the diagnosis. And this was because of suspicion about the delay in developmental milestones, gaze aversion, language delay, possible risk of autism, who was eventually referred to by the pediatrician to a clinical geneticist for a differential diagnosis of Fragile X. And at 2.5 received the diagnosis. This is before a formal diagnosis of autism would have, in fact, been allocated to this boy, given to this boy. But his little brother was only 2 weeks when he received a diagnosis, because he was already on his way when his elder brother received the diagnosis and was tested postnatally. So again, a case of within a family, but very common in the context of genetically identified differences that can be associated with a very early diagnosis before symptoms and difficulties manifest. And so I spent quite a lot of my time as part of my PhD trying to characterize the difficulties that these children encountered. But in that process, I became really interested in the differences between children with Fragile X, even those two boys in that picture, in terms of their prospect, their changes over time, and their changes in strengths and weaknesses. And there have been, again, some pioneers in looking at that work and looking at the influence of the environment on the individual differences that were measured in Fragile X. If you're interested in those, please, I recommend the work of David Hessel. So we embarked, therefore, in a longitudinal study. Longitudinal studies are what's necessary if you want to study individual differences and do so over time. It was a pretty big study. We did lots of things. The parents and the kids were so engaged, and we're so grateful again for their commitment to many cognitive tasks, as well as multiple other measures of their behavior. And again, in the interest of time, I won't spell out all of our findings. Many of them are published, so I recommend reading some of the work of my colleagues, and Eleanor Long and Vic Cole led much of that work. But for the purpose of this talk, what I want to focus on in particular are the individual differences, the variability. Again, because this is a common theme that we've seen in ADHD and reappears in the context of Fragile X. Now, a reminder, for those of you who don't work with Fragile X, it's monogenic in nature. So variability is very interesting from that point of view, and multiply determined probably by other aspects of the genetic background, by environmental influences, and by change over time. The graph here represents that element of variability. Light gray lines are individual boys. The dark black line is the trajectory for the group. On one particular measure, this is a measure of intellectual quotient, the Lighter International Performance Scale, but in fact we saw the element of variability at the onset and over time for pretty much all the measures we obtained. For example, highlighted an average IQ of 63, but pretty big range of variability from the floor on our IQ measure, 40, all the way to 109, which is within a normal range for an IQ measure. And similarly, in terms of ADHD symptoms, 48 score on the measure that we obtained is pretty normal. 80 is the maximum one can score on that measure, and equally for autism spectrum symptoms. I won't belabor the point, but even if you use a very gross cutoff measure of being above or below risk for ADHD, autism, what did we see in the group? Well, we saw a pretty even split of children above cutoff for ADHD alone, for ASD alone, for both, or in fact none. So this illustrates again the point of variability across this group with an identified monogenic high risk factor that can be diagnosed very early in life and can be tracked over time as well. And this is what Brianna did, in fact, with our longitudinal data. So some highlights of what she did. She looked at ADHD symptoms over the 3 time points that the study continued in, autism symptoms and ADHD symptoms, as well as IQ measured at multiple time points, and sanity check. These measures across time points related to each other pretty strongly. So ADHD symptoms at time 1 were strongly related to ADHD symptoms at time 2 or time 3. But there were also really interesting cross-symptom relationships with ADHD symptoms early predicting later autism symptoms, which is not something that had been reported before in Fragile X. And we have some really interesting mechanistic suggestions as to why we might see that happen in our sample. So from ADHD to autism and not the other way around. Again, if you're interested in that point in particular, please ask me later. And those relationships held even when we controlled for intellectual disability, so individual differences in IQ in our sample. So altogether then, to close off the second part of the talk, children diagnosed early in life with high genetic risk highlight many really interesting conclusions. First of all, because of their early diagnosis, they both provide the clinical imperative, a requirement for understanding them early, and the scientific opportunity to study ADHD symptoms as they emerge over time. What protects some children from those ADHD symptoms and what makes them bigger and more severe in other children with that same condition? Now, we didn't have time to touch on this point, but actually there's a high degree of methods development required for getting at cognitive underpinnings of those symptoms in children who have a lot of difficulties, so find standardized tests really challenging. And equally, getting at the neurocorrelates of those differences can be a real challenge. There aren't very many. The studies that have attempted imaging in Fragile X have had to overcome big difficulties of lying in a very noisy environment and staying very still for a long period of time. So it's not trivial to think about looking at neurocorrelates in populations like this one. But all of this work has focused on their risk being high in genetic terms. We've actually been very interested in looking at environmental moderators, but I don't have time to tell you about those in this context. What I wanted to close on, though, and then leave the rest of the time for discussion, is new work that doesn't focus on genetic risk at all and doesn't directly focus on ADHD either, but that does focus on definitely neurodiversity and diversity when thinking about attention and control in the context of environmental risk, poverty, and socioeconomic risk. This to me is very new work. So I just want to highlight that there are around you some great colleagues. Kathy Draper and Kaylee Cook were based at UCT and they're now at WITS, but led the work that brought us to work together in South Africa. Now, this is what I'm used to dealing with in terms of very early learning environments of children in the United Kingdom. This is your typical preschool classroom. With kids who are paying attention to the lesson, and my favorite kid in the picture, who's not, is really looking instead at the person who's taking a photo. And Kathy made me really think about how this environment could be different, but also similar to the environment she's used to. And so she brought us all along, first for a workshop and then for a study to Mpumalanga province, so Agincourt in the north of South Africa, rural communities. And here's just some photos that we took with permission from the carers in nurseries settings, where the type of environmental stimulation and manipulatives surrounding the children are really quite different. Interestingly, the learning activities are quite common. In both cases, this was a music lesson and they were singing, the children were singing. But also in this picture, you can see that there are some children who are really paying attention and there are some who are not. So it's commonalities as well as differences. And as a follow-up to that visit, then we were very interested in trying to understand cognitive characteristics of the children in these different settings. We also recruited a sample of children in Soweto, so both rural and urban South African children, of course, with very different and varying contextual factors associated with their development. we compared their cognitive performance to a pretty large sample of preschoolers in Australia. Stephen Howard, who's presented here, led in developing a standardization battery of executive function tasks and collected lots and lots of data on Australian preschoolers of varying socioeconomic status. And the tasks are all friendly. In the context of the South African study, they were translated in the home language of the children who were tested, had very intuitive and child-friendly instructions. They involved dealing with touchscreens, so we didn't know how children in the sample in South Africa would deal with this setup, especially given that they were from low income and in some cases really rural communities, but actually loved it. So the games involved rabbits, boats, ants, and sharks and fish. We can talk about them again later if you want, because what I want to close with are the initial findings of that study that we're now about to hopefully replicate and follow up. So this graph is pretty complicated, so I'm going to take you through it a little bit at a time. So in clusters are the measures from one of the tasks on the previous slide that was looking at working memory, then a task that was looking at inhibition, and a task that was looking at cognitive flexibility. Again, if you want the details, we'll return to those. And then the children are all split in quintiles in terms of their socioeconomic status. So the poorest children in Australia and South Africa are indicated as quintile 1 children, and the less poor, so richer children, are indicated as quintile 5, both in Australia and in South Africa. So those are the coloring. The bars have low SES, low socioeconomic status on this side, and high SES on that side. And one key point to keep in mind, and in fact the statistics hold as well as the qualitative observation of the graph, is that there is an SES gradient in our data across Australia and South Africa. The poorer children show weaker executive skills across both countries. And you see that represented by the arrows. But there's also a very interesting finding that we'd not expected at all, and we're really interested in replicating and understanding better, which is that the South African children, in absolute terms, outperform the Australian groups of comparable SES. In fact, we don't really have very comparable SES because the poorest children in Australia were richer than the poorest children in South Africa. We don't have very good comparability across that. But if you look across the three measures, in the dark blue are kids from South Africa, in the lighter blue are children from Australia, and the children in South Africa outperform, so are better in their executive skills than the Australian children. And that is in fact the same across SES quintiles. We see it consistently the case across the board. So very interested in understanding both the gradient, so poorer children doing less well within a country, but also the differences across countries. And we've attempted to collect some imaging data by using EEG because it's a little bit more portable than MRI, although I'm really counting on the people coming up with portable MEG, really making it cheap so we can have access to it. We used some portable EEG. The data collection was complicated. If there are methods, people in the audience who want to hear about how we dealt with braids and portable EEG, please ask. But interestingly, for this follow-up study that's only just started, but in fact is now on hold because of COVID, we're really interested in studying longitudinal change and studying, first of all, testing for replication for the previous findings, and then looking at both executive functions, attention, skills, and outcomes that are relevant to early schooling for those children. Okay, so taking stock, and I think I'm actually possibly on time. Good. That's not normally the case with me, so phew. But I wanted to give enough time to Kirsty to speak. So just the highlights. In the context of ADHD, we've seen that there are clear structural differences between kids with ADHD and kids without. There are also interesting effects of extrinsic motivators on functional engagement of kids with ADHD's brain. And there are individual differences as well that we need to understand within the group of kids with ADHD. So we really need to think about neurodiversity and extrinsic motivators more. This raises some challenges as well as opportunities with the need to replicate our data, the need to collect larger samples, the need to collect longitudinal data, and the need to have information on differential responses to interventions. But I've also linked in, again, I've also focused on ADHD in the context of children who may receive a diagnosis that puts them at very high risk for ADHD, but way before a diagnosis is allocated. And those are intriguing populations, I think, because they allow us really to delve into the precursors of ADHD symptoms as they develop, but potentially protective factors. Who are the children who were at risk, but did not then, as young children, really develop those inattentive and hyperactive symptoms? Understanding those children, the resilient children, is just as important as understanding the children who have a lot of difficulties. The resilient children might give us insight into what might help everyone. And so genetic syndromes are developmental in nature. They offer really interesting opportunities, but require longitudinal designs, and also state-of-the-art methods that get their cognition when some standardized measures don't. We need large samples, and this is ultimately what we need to do to investigate mechanisms of risk and protection. And to close then, we've only just started dabbling with environmental risk, and I'm really excited to hear from much better experts than I am in this area. I've been intrigued by the findings of that first study, and I'm raring to find out whether they replicate or not. But also would like to understand the mechanisms of both strengths and weaknesses, and I don't think we have a full understanding of that. And I close with thanking all the many, many, many people who are behind the good things I've told you about. Any of the lack of clarity is my own fault. Thank you very much. That's it. Oh, and I'll stop sharing. These are my kids, by the way, who have been gracious and have not showed up at the talk.
Thank you so much, Gaia, for a really, really interesting talk. I really, really enjoyed it. So now I'd like to open up to everybody for questions. So how we're going to run this, if you'd like to either use the raise your hand function on Zoom, or if you'd like to type into the chat that you have a question, and then I can call on you to ask it. So you can unmute and ask. That might be the easiest way to do things. So I'm sure people have loads of questions. Just give me a second.
There's also a virtual pub discussion later, isn't that right?
Exactly.
I want to make sure I don't take time from Kirsty, so we can do virtual chat later. Virtual pub.
Okay. And okay, there's one question here from Julie, but before I call on her to ask the question, just in relation to the virtual pub chat, that won't be streamed live on YouTube, so people can show their faces and don't have to be worried about being broadcasted on the internet. So Julie, if you'd like to unmute and ask your question.
Yeah, hi. Thank you, Professor Scerif, for your talk. My name is Julie. I'm based at the Perinatal Imaging Department at KCL. I study the effect of maternal obesity on developmental outcome in a child. Yes. And we see that many offspring born of obese mothers have higher risk of ADHD and autism. I've studied lots of the mechanisms, and because of your generous offer to the effects here, I was wondering whether you had thought about the differences, for example, in diet that you'd find in the poorer South African or Australians, because Australians would probably tend to go for the Western diet, high-fat diet, if they're on the poor scale.
That's a great question.
On the healthier scale of that diet, possibly.
That's a great question. In fact, my colleague, Kathy Draper, is very interested in this question. She's interested in physical activity, but also in nutrition. So in that first study, because this was our first attempt to work together, we didn't collect very detailed data on parental practices, nutrition, but we are going to for this new study. And she's very heavily involved. I mean, I'll very happily send you the details offline. Kathy is very heavily involved in a large-scale new cohort study in Johannesburg, I believe, trying to collect data around mums and very young children and babies, and also a higher incidence of obesity and nutritional differences, as well as physical activity differences in South African mums and baby pairs. So I don't have an answer to your question. It's a great question, and Kathy is really interested in it.
I mean, so only the second thing that comes to mind, apart from the diet, because obviously they are my cohort of interest, there would be the prevalence, for example, of diabetes in those groups. So I'm always going to think about the antenatal effects. So you have the diabetes that might have the same outcomes, and depression, so psychopathology. So these three factors, the metabolic, dietary, and psychopathological effects antenatal, but
Yes.
the postnatal effect could have like a multiple hits. So these are kind of the things that you might
Absolutely.
want to look into wording.
Yes, absolutely. And I mean, again, I don't know if you know other teams here in Oxford, there's very big interest in perinatal and postnatal depression, both maternal and paternal depression, and how those might be risk pathways that may be larger as risk pathways in individuals who are at high socioeconomic risk. I mean, the interesting thing is that probably nutrition has an effect across the board, but it may be that is also characteristic of low-income contexts more, and definitely that needs characterizing for sure. We are going to be doing that in the context of the new South African study, looking at parental well-being, mental health, as well as nutrition and physical activity.
Thank you. Look forward to reading it. Thank you.
Yeah, well, I mean, we're so excited, but unfortunately we will stop because of COVID. In a way, the silver lining is that we're actually really thinking about all the measures that matter, all the things we should be knowing about these children and families as we recruit them again. So thank you for that.
Yeah, thanks.
Thank you.
Thanks, Julie. Gaia, we have another question from Richard Berman. Richard, would you like to ask a question?
Yes, yeah, go for it.
Cool. Thank you very much for a really interesting talk. I've just got a question around the structural differences that you saw in the ADHD patients, and when you start seeing those. I don't know the literature at all, really, but when would one expect to start seeing changes in the circuitry on imaging? And a follow-on question to that is, in your patients with Fragile X, do you see a similar type of structural change in the patients that developed ADHD compared to those that didn't, even though they both have Fragile X?
That's a great question. And let's take the ADHD part first. Now, there are now some really interesting longitudinal studies that look at mainly NIMH based, so North American and large scale studies that look at longitudinal differences and how they hold over time in ADHD. Of course, they're in kids who are diagnosed, and who have entered into studies. So we don't really know about any children who maybe had earlier inattentive and hyperactive symptoms but didn't reach diagnostic cutoff, or who are not willing to take part in imaging studies. But those areas, the ones that we saw in our study, are also areas and circuits that are implicated by the longitudinal studies. And those differences don't seem to resolve. So there is some really good, I can definitely direct you to that work, some really cool work that shows some pretty consistent differences. We don't know pre-diagnostically or early in life, because that's not, you'd need a population study to ask that question of ADHD. And I don't think that's been done, but if anybody knows of that, then please tell me. In the context of Fragile X, your question is super interesting also, because what has been done is functional and structural imaging work on the frontostriatal circuitry. And what has been found is that there are differences between kids and adolescents and women with Fragile X in those classical areas. But the question that has not been asked yet, as far as I know, is about the differences within a group with Fragile X. So the replication of differences in volumes and differences in functional activation of frontostriatal circuitry in Fragile X compared to controls, definitely there. And there's work by Alan Rees and his team at Stanford, I believe, that does that. But what's not been asked is, what are the differences between kids with Fragile X with ADHD symptoms that are very severe and those without? As far as I know, that's not been studied.
Cool. Thank you.
Thank you. Oh, there's a question from Anne Dowker, is that right?
Yeah, so we might take this question from Anne and then move on to Kirsty's talk. And anybody else with questions, come along to the chat.
Thank you. Thank you.
Anne would like to unmute her microphone and ask it herself, or we can just read it from the chat.
Yes, my question was that while I'm not an expert on the subject, I understand that some people have questioned whether reduced deferment of gratification in poorer people may not always be a deficit, but may be an adaptation to circumstances, that if you only have access to food or luxuries intermittently and perhaps don't have a fridge or other preservation facilities, it might make sense to use things when you have them. And I was wondering whether this could be relevant to some of the national differences.
Yeah, it's a great question again. I mean, well, not unfortunately, but interestingly, none of our executive tasks were delay of gratification tasks. So, I mean, so yes, so absolutely. I think, there's a lot of work on delay of gratification in food insecure environments. And therefore, what might seem as come across as inhibitory deficits, not being able to wait and not delay, as in fact being the right thing to do if you're in a food insecure environment. In our food insecure environments, the kids we saw were better at inhibiting than in Australia. So that's interesting, because I don't know, I really want to see this, whether what we see is replicated and what the potential underpinnings of that might be. There may be an element of greater independence in the children we saw in South Africa. Many of the kids we were seeing, oh, sorry, there we go. Many of the children we were seeing were going to school for many miles, bringing their younger sibling with them. And that's not something that's very common, even in low-income communities in Australia, for example, right? So there may be some really interesting contextual and environmental differences there.
Thanks very much.
Thank you very much, Amy. Thank you.
Thanks again, Gaia.
I'm going to mute myself now. Okay.
Okay, thank you. So I'll pass over to Aaron now. You'll introduce Donald.
Yes. Thanks, Prof. Scerif. We appreciate that. That was a fantastic talk. And thank you everyone for the questions. And just a reminder that if you still have a burning question, you can ask it in the virtual pub chat later this afternoon. So let me just introduce Prof. Kirsty Donald. In Cape Town this year, UCT established a brand new Neuroscience Institute, which encompasses the broad spectrum of basic and clinical neurosciences. The vice head of this new institute is Professor Kirsty Donald. She's a pediatric neurologist and the current head of the pediatric neurodevelopmental unit at Red Cross War Memorial Children's Hospital, which is the largest children's hospital in sub-Saharan Africa. Prof. Donald has a special interest in the effects of alcohol and HIV, as well as methamphetamines on the developing brain, as well as trying to identify broad psychosocial issues which impact on neurodevelopment, particularly in the South African context. Today, she'll be sharing some of the lessons she's taken from neuroimaging the very young high-risk brain. So without further ado, I'd like to welcome Professor Donald.
Thank you, Aaron and Tara, for this invitation. I'm very excited to speak to this group, not only to share the story of this cohort and some of the interesting lessons that we've learned from it. Certainly, we haven't finished learning all the lessons that we have, but I will give you a little bit of the background and a few snapshots of what we've learned so far. But because this is a group of people who are, or many of you are in the learning phase in finding your path in your careers and presumably you're interested in neuroscience, otherwise you wouldn't be here. And I think these ideas of being able to collaborate across the north-south divide and across the divide of high income and low income environments and have exactly the kind of conversations that we've just been having about why things might play out differently in different contexts is really exciting. So I'm very, very, very happy to be part of this and look forward to sharing our story with you. So let me try and find my talk. It's not giving it to me. I didn't open it up and go to the beginning and let me just try, sorry, muttering. See if that's better. Right, are we screen sharing? You should, at this point, yes.
Yes, though we can only see half your screen.
Okay. Let me just go on
now
to
now it's okay.
the view. Is that better? Can you all see it? Excellent.
That's perfect.
Yeah.
Okay. So for those of you who are wondering, this is a picture looking up at Devil's Peak from the roof of the new Neuroscience Institute, which Erin mentioned. So we're situated right. So those of you from South African context, you're going to have to just bear with me while I describe where we're from. So we're based at the tip of Africa, which, as you can see, is extremely far, much further south than England, and in fact Europe. Africa on maps is often represented as much smaller than it actually is historically, a sort of little squished continent, and in fact it represents a vast landmass with huge numbers of people in clusters, but also spread out in deeply poverty-stricken and rural areas. This study that I'm going to talk about today really represents a kind of peri-urban community, but gives one a little a signal to some of the psychological, socioeconomic, and environmental risk that these children face. There are very few places in Africa or really in the world, where one has access to populations of children who are growing up in these kind of high risk environments, and the resources and infrastructure to be able to study them with a level of sophisticated neuroscientific techniques such as MRI and genomic investigations, and South Africa is one of those areas. So we have a unique privilege in a way to be able to do these research studies, but also a huge responsibility to make sure that the money that we spend on studies in these environments have translational potential and are answering important questions that are important to the community, and are also important in terms of addressing the particular challenges to brain health in our context in high risk environments. So this is where we are on the map. Again, this is a picture of the Neuroscience Institute, and you can just you get an idea that this is Cape Town. It's a very urban cosmopolitan city at the tip of Africa, and we are relatively well resourced compared to many other areas in South Africa and across Africa. And this is in fact it's a stock shot. It's taken off the internet, but it is a picture from another region in South Africa, which is less than 500 kilometers away from Cape Town. So, this is deep rural poverty, where people don't have running water, don't have electricity in their houses, and you can see that the discrepancies that exist are huge. And while in the last century, the analogy that people use is that the water has risen, and that many millions of people have been brought out of extreme poverty, but at the same time the discrepancies the differences between the rich and the poor have grown. And our recent the recent COVID epidemic, I think because of its global reach, has really brought a whole lot of really salient lessons across the world, that the vulnerable are much more vulnerable in a crisis, and that sorry, I'm just checking that. Can you still hear me? Okay, sorry, the internet sometimes drops us here. And that as the world continues to go through the COVID pandemic, we're at the moment, and we come out of we go into the kind of economic slump that comes out of that the children who are in that lowest social quintile are going to be at highest risk from a food security perspective, from a social violence perspective, from schooling deprivation perspective, clothing, warmth, protection, all of those very basic things. And so these lessons that we are learning are we going to need to apply in spades when we think about how this feeds into interventions. Another way of looking at this is this is based on models. So we don't have very good hard data on how on prevalence of intellectual disability, or in fact any of the developmental disabilities across Africa, but based on sort of proxy measures, of which stunting is probably the most widely used. Africa is really overrepresented in terms of intellectual disability, and that is not because people have different potential here, but they definitely have different risk profiles, and so the secondary causes of intellectual disability are rife in our environment, and we're going to speak a little bit more to that in detail. Again, this is a fairly blurry shot, and this is taken off the dark web. It's a picture accessing the internet. And as you can see, Africa is really predominantly dark. We've got a little highlight around Johannesburg, which is our biggest city, and a little spark in Cape Town. But for the rest, the continent is very poorly connected in terms of Wi-Fi and data. And so while we gallop ahead, being able to do joint symposiums like this, if we're wanting to get information to people and have conversations with people where there isn't good connectivity, we need to be creative about what platforms you use, what data approaches we use, and it just requires a completely different way of thinking. So that's just something to bear in mind. But it also is probably a proxy for what kind of access the children in our environment are getting to schooling during the COVID pandemic. My children are sitting at home. They've each got a device. They are borrowed, but they are nevertheless were accessible within my family structure, my extended family structure. And they can go online for 3 hours at a time. They can go online together at the same time while they're in school. And what are the chances that people living not 50 kilometers away from us in informal settlements have the same kind of infrastructure? So there's just a lot of discrepancies that we need to be thinking about when we try and understand what's going to help in our environment. This is again based on models, and lots of things can models, but these are population projections into the future. And you can see that on the left is, sorry, I don't have the dates in clearly, but on the left is 2015, and you can see that African populations as a proportion of the global population is relatively modest. But as you approach 2100 over the next 80 years, the population in Africa comes to represent a much larger proportion of the global population. What does this mean? What does it mean when populations grow quickly? It means that, I mean, I'm not a public health person, but this much I know, that when you grow a population quickly, the greater proportion of the population is going to be the young people and children. So children will remain the major focus of Africa, and Africa will be the main focus of the world's children. And so it's crucially important to understand all the factors that contribute to the healthy child's brain development and function in our context globally, but particularly in our context. As I said, in 2050, of Africa's projected 2.9 billion population, so on this continent, 1 billion, so 35% will be children under 18. And children in Africa will account for nearly 40% of the world's children by then, and rise to a staggering 49% of the world's children by 2100. So this means that we have a very special responsibility as neuroscientists and clinician scientists in Africa who are interested in these areas to do a good job. And not only do a good job, but train and capacitate other people to be able to lead in these areas, so that we make sure that the questions that we are asking of our research, in our research, is answering the important questions for these children. As Gaia has already talked about, there are many things which feed into how you do in your life. I'm not going to go into all of these, but you can see that there's your genetic endowment, which has huge variation, and particularly in African populations because of our original genetic history. We've got environmental factors. We've got factors that may be impacted on over time by different elements of these things. We've got epigenetic factors as well. And then we've got sort of biological environmental factors such as excess inflammation. So I'm going to talk about a few of these as we go along. So these cascades of different elements impacting on a child's brain growth and development act within periods of development, but also across periods of development and across generations. So this obviously makes it particularly complex to study, and it's helpful to have longitudinal studies which include more than one generation. Obviously, both positive and negative cascades may be evident, and as Gaia also mentioned, identifying those that act positively, or in children that react positively within a high-risk environment, is as critical as identifying the risk, because that group are the ones that one wants to try and understand what makes them be able to be fine in this environment, what makes them thrive, and how can we try and support those kind of features for other children in the same environment. Obviously, cascades may be disrupted at later points in the life cycle as well. So it's not all about the early years, but the early years are probably one's opportunity for having the greatest impact. But what I would say is that the story doesn't end there, that you need to focus on early child development and interventions at that time, and factors which feed into that time, but you have to actually keep intervening. You have to identify what are the threads that put children at risk, and start early, but keep scaffolding children through their lifespan, those at risk. I think I've spoken to this in enough detail, and because I'm not focusing on middle childhood, it was really just to mention that there are a lot of things in middle childhood, and particularly in adolescence, as people become, as young women go into childbearing age again, that we need to be closing the loop, and we need to be thinking across these risk factors across with a lifespan approach, not just in our little areas of age-specific expertise. So that's where you want to phone a friend. You want to have collaborative groups of people who are able to think together about the problems, and not just operate in one's little silo. So a little bit of background to why, what we're doing in this cohort, and with a particular focus on magnetic resonance imaging. So MRI in young children, I'm preaching to the converted in this audience. It's a valuable tool for investigating early brain development and the neurobiological mechanisms underlying developmental risk and resilience in a high-risk context. Beyond early infancy, pediatric MRI imaging is challenging due to motion, particularly in children under 5 years. So in the neonates, you can, they're very sleepy little beings, and you can feed them and wrap them in a fluffy blanket, and often they will go to sleep. It's a relatively predictable sequence. They don't always sleep through a long MRI scan, but generally you can get them to sleep. Anyone who's tried to get a 2-year-old to cooperate with with even a short task, never mind falling asleep in a scary environment, and going into an MRI machine when they've never been into a hospital before, would know that is really dragon territory. But what I'm going to tell you about today is how we have managed to do that. In the research context. So obviously in the clinical context, you're able to use sedation and anesthetic because it is justified for clinical purposes. But in the research environment, one can't do that because the risk isn't justified ethically. So what are the questions we're asking, and how did we go about doing it? The question we were asking is, well, isn't pediatric multimodal MRI in these very young children feasible without sedation in our setting, especially because children really don't have any experience of big hospital environments and more sophisticated infrastructure, and often very afraid. What factors affect brain development in this high-risk environment? Are there some factors which seem particularly important? And then how do these factors affect the very young brain? So the areas that we are focusing on generally in the Neuroscience Institute, but in particular in this cohort, we are looking at infection in the brain. And the little bit I'm going to talk to you about today is children who have HIV exposure without infection. So with the rollout of antiretrovirals, more broadly in South Africa, most children, if their mums are infected, their mums go on to antiretrovirals in pregnancy, and they don't get HIV infection. A few still do, and some mums fall through the cracks, obviously, but largely speaking, we have a very low mother-to-child transmission rate in the country. So this means that there's a growing population of children who are born to mums who have HIV, but who are not infected themselves, but they're exposed to the mums' ill health and pro-inflammatory environment, immune dysregulation, and of course the antiretroviral drugs in pregnancy. And some of the questions are, what does that do to the developing brain? Are there things that we need to worry about, and what are they? The other areas, obviously, are violence exposure, trauma and stress, substance use, in particular alcohol use is very prevalent in our region, but also methamphetamine, or TIC as it's known in our environment, maternal mental health, and then of course a brain injury as well. So this is a slide which is courtesy of Catherine Wedderburn. I think she is on the call. She's a PhD student working on this cohort, who's doing her PhD on the HIV exposed uninfected children. So she's played a huge role in being able to get the imaging done on these 2 to 3 year olds. And this slide illustrates the rising number of children in our environment, because the epidemic of HIV is in sub-Saharan Africa. You can see that there's a rising millions of children who are HIV exposed, and increasingly numbers of children who are HIV exposed who have been exposed to antiretrovirals in pregnancy. So this becomes an important question to answer, not only whether the antiretrovirals make a difference, but also which of the antiretrovirals is best for the developing brain. And these are questions that we probably urgently need to answer over the next little while, so that we can make sure that the mums in pregnancy go on to the best regimes to prevent the HIV, but also to protect the developing brain. So we are exploring some of these questions within the Drakenstein Child Health Study. It's a population-based birth cohort study which has the broad aim to investigate the early life determinants of child health. The methods are that it's a multi-year birth cohort study. It was started in 2012 with pregnant women. Approximately 1,000, so just over 1,000 were recruited in pregnancy, in the second trimester of pregnancy, and they're being followed up into middle childhood. At the moment, the oldest children are just about to turn 8 and the youngest are still 5. And so we're following them up prospectively, hopefully even longer, but you know, that always depends on funding. So this is these are a few pictures which give you a bit of a snapshot of the kind of home environments that children in this cohort live in, and they range from families who live in blocks of flats here with running water and electricity and really kind of reasonable basic amenities, to children who are growing up in very informal housing with very, very few resources and plenty of food insecurity and other issues. How did we look at development? So with a focus on the measures that we used. So this is because this is a broad child health study, there's a absolute rich data set of set of data on both the kind of biological health measures, psychosocial risk and exposure, and so I'm just focusing on the outcomes that we are looking at with respect to brain right now. So for the very young children, so under 3, we looked at the Bailey scales of infant development, which most of you will have been exposed to as one of the gold standard developmental assessment tools, which has a direct element of, so it's not just a questionnaire, it's a direct assessment of the child's development across different domains, more broadly across cognition, but also language, motor, and socio-emotional development. We also did neuroimaging. So we did this we have a 3T research scanner at the Cape University's Brain Imaging Center, which is basically attached to the Neuroscience Institute. And we did this in a subgroup of the children with some exclusion criteria, obviously. The different age points that we've done it at are, we imaged about 240 infants in the neonatal period, so between 3 and 6 weeks. And then we followed up a similar number of children at 2 to 3 years of age. And at both, and we are now imaging them again at 6 when they are more reliably able to cooperate a little bit. But at all of these time points, we have in fact done 4 different modalities. So we've looked at structural MRI with attempts to do T1 and T2 sequences. We've looked at diffusion tensor imaging, which measures the white matter microstructure in the brain. We've looked at resting state functional connectivity, which looks at blood flow utilization during rest, which obviously, because the children were sleeping and in the 6-year-olds lying quietly, they're not doing particular tasks. You can't really ask people when they're sleeping to do tasks. And then magnetic resonance spectroscopy, which focuses on a particular, you have to choose a particular region of the brain that you're interested in, and then we'll read off a sort of a chemical signal of different neurometabolites. So this was the setup, which we obviously tried to make as child friendly as possible. Judy Gate was one of our research assistants on the study, and she was a wonder at getting the children to sleep. Firm and warm is what you need for 2-year-olds. Sometimes the children are happy to go into the scanner, but most of the time they needed to be asleep. So we just show this picture because it was actually possible. A 3-year-old did go into the scanner willingly, but she was the most incredible 3-year-old. Catherine took this picture, and it is this little girl is our absolute poster child. She did a fantastic job, but the rest of them needed to be asleep. In terms of our neuroimaging enrollment, I've been through, just to kind of give you a picture of how this all fits into the umbrella study. So we had 1,100 subgroup of about 230 who we imaged and in the neonatal period, and we obviously tried to image the same children again at 2 to 3, although we don't have complete overlap. And then, sorry, I've gone backwards. And then some of the children didn't sleep, as you might imagine. We had to be quite creative. We got permission to scan at night in the evening when children were due to go to bed, and we brought them in from their homes. We collected them in a taxi, brought them in, gave them a warm meal, and then encouraged their mum to get them to sleep. And in our environment, either that was on their mum's lap or on their mum's back in a blanket. And amazingly, many did sleep. And I give huge credit to Catherine in this regard. She was very persistent in getting this right and getting our team logistics right, so that we had such a high success rate. And this is really comparable to other centers in the world. So we managed to get nearly 80% of the children to have a successful scan out of the 2 to 3-year-olds, which is really kind of phenomenal. And but obviously that wasn't across all 4 sequences. So we got at least a structural scan on 77%, and then it goes down for the noisier, more annoying sequences, which tended to wake the children up. So here, this gives you a little bit of an outline of the different success rates across the sequences. And so on average, our children were about 2.5 when we scanned them, between 2.5 and 3. Slightly more of them were boys. And as you can see from their household income, so to give you an idea, that the exchange rate at the moment with British pounds is about 1 to 20. So 1,000 rand is about 50 pounds. And so if you think of living on 50 pounds a month, that's not very much money. And that's a third of our children, of our families, are living on less than 50 pounds a month. That really is extreme poverty. Many of our mums have at least some primary education, and some have some secondary education, but not very many children. So 2/3 of mums did not finish secondary school. So that is quite a high proportion of children, of mums who haven't finished school. Only 1/3 of mums were employed, and they were mostly mums in their 20s. To give you an idea of the children, on average, the mean raw score with standard deviation on the Baileys in the cognitive area was 86, which is about 1 standard deviation below the American means. And this is widely described in tools which are not normed to lower middle income country population. So this, just to bear that in mind. And likewise with language development in a similar range. We looked at what factors might have influenced, or what factors seem to be associated with better scan outcome. Ooh, what's happened here? And predictably, there didn't seem to be a huge any major associations, because it really is more a kind of behavioral thing. But it was reassuring. what happened once you've got the scan, that's not the end of the story. So this is again a beautiful slide that Catherine has put together, which just shows all the stuff that has to happen once you actually have the scan data. So once you have the data, you have to check it, you have to process it, and then of course you have to analyze it, and then you have to put it together with understanding what the functional outcomes are. So it's imaging is quite a challenging modality to look at, but it's worth it because you get a really good picture of what's actually going on in terms of these children's brains. So this is just a pretty picture of Cape Town on a winter sunset, just to distract you briefly, but different areas of interest that we've investigated. So this shows the structural differences between the children, the babies who were exposed to alcohol in pregnancy. And just to give you an idea, the mums were not necessarily heavy drinkers. So the mums that we included in the alcohol exposure group were either moderate or heavy drinkers. So they either had at least 2 drinks a week during the second trimester of pregnancy, which is not really, I mean, the British Medical Association recommended a drink a day in pregnancy up till about 10 years ago. So 2 drinks a week is not exactly heavy drinking, but nevertheless, in this group, the deep gray matter seems to have been primarily affected, so the subcortical gray in terms of volume. And the interesting thing is that when we look at a number of the different risk areas, many of them focus on this region. So obviously it's a very highly metabolically active region, and obviously deeply involved in the kind of early network formations of the tasks of early childhood and early infancy. And so it's probably not surprising that these areas seem to come up as being affected in early infancy. And this was associated with neurodevelopmental outcomes as well. When we looked at both structural and functional networks using the fMRI, the alcohol exposed groups had very much less organized networks. I think that it's still again, this is the early brain, the neonatal brain is only a couple of weeks out of the in utero environment, and so it's not always easy to tell which things are going to are still a sort of hangover of their exposure in pregnancy and are going to settle down with time, and which things are going to persist. But if you don't describe the stuff that happens in those, or that is identifiable in those early weeks as a result of these risk factors, it's difficult to tell later on, because of all the other environmental risk, what is actually causing the problem. Whereas if you've seen a problem early on and the problem later on is connected to it, it really gives you a bit of a thread for which pathways seem to be most vulnerable to these particular risk factors. So it's relevant to look early, even if you don't have a very clear idea of that particular area of significance. What about the HIV exposed group? We have in fact looked at both the structural outcomes and the diffusion tensor imaging, or the metrics for this group, for this age point. And there were changes in the white matter microstructure, particularly in the cerebellar peduncles, but also in the kind of major association tracts between the children who were HIV exposed and those who were not exposed. And Catherine's about to submit her first paper on the imaging outcomes in the neonatal, on the structural analyses, which show again that the deep grey matter seems to be mostly affected in that group. So we've started analyzing the 2-year-olds as well. This is still, it has taken us a while to get it all together. And this paper has been published, which describes the success rate in the 2-year-olds and associations with cognitive outcomes in this group. In the HIV-exposed uninfected cohort across development, we've described that children who are HIV-exposed had worse language outcomes at 2 years on the Bailey scales of infant development compared to those unexposed. And in Catherine's preliminary analysis on the structural imaging at 2 years in this group, it looks like this, that the areas that are affected, the cortical maturation appears to be affected in areas which are associated with language development. So this is a really important story, I think, that is coming out of this group. This comes from the work of Cesc Bertram-Cobo, who's on the right here. He's an MSc student who Catherine Francis Robertson and myself supervised. And he recently looked at the MRS data in the children who are HIV exposed and uninfected. So just to recap, MRS looks at, is magnetic resonance spectroscopy, and looks at neurometabolites in particular regions. The regions we looked at were the parietal gray and parietal white matter. To be completely honest, the areas we chose were entirely pragmatic, because in the neonatal period, the only bit of the brain that you can be sure you're going to hit is the parietal gray and the parietal white. Any of the subcortical structures are too small, and the gray-white differentiation is too poor at that age to be sure of anything else. So that's what we went with, and so obviously we followed up with that. So this is looking at the MRS in the 2-year-olds, and just to give you the background of what these neurometabolites mean, if you the spectrum that we have looked at is NAA, which is a kind of traditionally looks at a pattern for neuronal integrity. Myoinositol is the pattern for inflammation. Total choline is is supposed to be a pattern for membrane maturation, and glutamate for neuronal maturation, and it was also affected sometimes in inflammation and substance exposure. And so, interestingly, in the HIV-exposed group, in both adjusted and unadjusted analyses, the inflammatory pattern seems to be, the signal seems to be coming out in the inflammatory pattern, which is kind of what one would expect. And so this really speaks to how the hypothesis around inflammation and immune dysregulation is probably a really important part of the story for how HIV exposure, but even in those uninfected, impacts brain development in these children. So in summary, neuroimaging is a powerful tool for exploring mechanisms behind developmental risk and delay. This particular cohort provides a unique opportunity to investigate neurodevelopment and neuroanatomy longitudinally. We have 180 children successfully scanned in the neonatal period and at 2, and we've got about half of them scanned again at 6 and are on pause, but we hope to start again before the end of the year. It's the first MRI study of children in this age group without sedation in a sub-Saharan African setting to investigate neurodevelopment. So I think that these findings are really important and demonstrates the feasibility of neuroimaging young children during natural sleep. The morphological changes in heteromodal association regions associate with cognitive and language development at this young age. And this helps us understand the interplay between brain structure and function during brain maturation. I'd like to acknowledge these. I'm sure I've left people off this list, but there are, this is one of those studies which has, has people doing the assessments on the ground, and people coordinating the study, and people in the imaging scanner, and people processing the imaging, people giving advice from the outside. It's a huge team effort. And so just thank you to everybody who has taken part in this. My daughter photobombing in our end of 2-year-olds scanning celebration supper. And this is the Neuroscience Institute opening evening, which amazingly was held in the first half of March 2020, literally a week before the first COVID case landed in Cape Town. It's very hard to imagine being in a group of people this large right now. And it just is an indication of how much the world has changed and how quickly, but we still hope that you will come and visit us there sometime soon. There's a lot of exciting work going on here and a lot of people to collaborate with. And with that, thank you.
Thank you so much, Prof. Donald. Thanks for that. We can now open the floor to some audience questions. You can leave the questions in the chat or just put your video on and start speaking.
Don't be shy.
Okay, so I've got a question. Can you hear me?
Yes.
Yeah. No, so that's not my my area of expertise, but I was wondering why you see changes in amygdala. I might have missed it, but so why do you think is it relevant to the problem you were analyzing?
In the alcohol exposure group?
Yeah.
So it wasn't only the amygdala, it was the amygdala and the hippocampus.
Right.
And I think, it's very difficult to make firm conclusions at that age, because really the kind of the tasks of infancy are very much in the kind of sensory motor domain. But I guess there's a lot the emotional content and memory and the networks around emotion, around regulation, are very embedded in that region. And so one would assume that those play a role. And what has been described as children with alcohol exposure can be quite irritable. And so there isn't really a withdrawal syndrome, but heavily alcohol exposed children can be quite irritable. And so that may in part be a reflection. But it's, I think, increasingly you're making sense of particular areas and saying that area relates to that function, especially in the very immature brain, where things are less differentiated out. I think is maybe, I mean, we all do it because that's the kind of paradigm that how we've approached it in adult neuroimaging studies, but I think it's probably less useful in children, and it's more useful to think about it as part of a network, and also in its longitudinal relevance. Does that answer your question?
Yeah, okay, thank you. Thank you very much also for the great talk, and both scientifically and also from the human point of view.
Okay.
Thank you.
I see there are two questions in the chat. Anne, would you like to ask the question live? You're muted at the moment.
Yes. I think I'm unmuted now. I was wondering whether this is the first study that's been done on HIV exposed but uninfected children's language and cognitive development, or whether there's any data from other countries.
So there are other data. There's a big study in Zimbabwe looking at a group at of a similar size. So, you know, when we describe the, or when we report on the imaging, it's on a small group, but the development is across the entire cohort, which is, you know, 900 to 1,000 children. The Zimbabwe cohort also looked at development and also reported language effects at age 2. And in fact, there's another cohort in Cape Town, a different cohort, which has described some cognitive findings in the children at 1. But there are also studies which have not found differences, and so I think increasingly, I think if you take the studies together, and there now are a fair number that are in the post-ART era, because the early studies were when the mums were not on ARVs and they were sick, and so obviously those kids do worse, but in the post-ART era, when the mums are relatively well, they do better. And when we looked at whether their immunological status related to the outcome, it does seem to be associated. So better immunological status, kids. If the mums have a better immunological status, the kids do better. So it kind of fits in with the picture, really. But I would say that language, cognition to a lesser extent, but language seems to be the thing which is coming up most consistently in the studies that find something. Does that answer your question? So the other questions I'm reading here are the neonates who showed the biggest effects in those subcortical areas, also the ones with the poorest language at 2. I don't know the answer to that question. Catherine, do you know the answer to that question? Are you here? Do you want to speak up? No? Okay.
I
Yes.
don't think we've looked at that yet, but I think that would be really interesting.
Yeah, it would be. I mean, we've looked at, Catherine's looking at the structure in the age, in the image 2-year-olds and their developmental outcome, and that's linked to language, but it's different. You know, obviously, it's a different relationship if you're looking at the early findings.
Yeah, but it's really, I mean, the data at each time point are amazing, very interesting. And then the really interesting additional steps is to look at relationships from neonatal parameters to the later ones. And maybe they are the same risky group, but I'm not sure. Maybe might not be the same mechanisms.
No, it might
really interesting.
Yeah. I mean, one also needs to take into account that children who are growing up in families where there are family members who are HIV infected, they have other psychosocial risks. So to a large extent, we've tried to control for those and make sure that the groups are as similar as possible. But obviously there are other things which may be playing into that over time. Did you measure maternal?
Look in a subgroup in Anker's paper. We did look in the subgroup and find an association, but it would be nice to look at it in the whole cohort.
Yeah, exactly. For sure. Okay, so the next one's from Oliver. Is alcohol or HIV exposure correlated with SES or other demographic variables? Have you tried to control? We have controlled for SES. And we have controlled for other demographic variables. And I think it's one of the big strengths of the study is that we have those. We have smoking and alcohol and other important things, intimate partner violence, maternal depression. We have all of that data, and we make sure that the groups are balanced in that region, in across those exposure categories, and do sensitivity analyses and control for them. So I think that we're quite well placed to be able to make sure that what we're saying we believe to be true, and not just some effect or some other confounding factor, but you can never be sure, obviously. So we'd obviously be delighted if other people can replicate some of these findings. So Julie is asking, did you measure maternal psych outcomes and IQ? So the mums have had a number of, they've had repeated measures of depression using the Beck's depression, the BDI, and then after birth the EPDS as well. So we do have longitudinal trajectories of maternal depression as well. And we also have maternal IQ and sort of a more extended neurocognitive battery in a proportion of the mums, and the mini in a proportion of mums. But most of that was done quite early on. And we haven't used the maternal psych. We haven't used the maternal IQ outcomes. We've just used maternal education in our analyses, because they weren't exactly the same mums as we have imaging on the children for, if you see what I mean. So that's just sort of unfortunate. So were the results similar in boys and girls? So they to an extent, when we looked at the data that I didn't present on MRS in the neonates for the alcohol exposed group, was that there seemed to be changes in the boys and not the girls. And in our big analysis of the neurodevelopment across the whole cohort at age 2, so just at the general look, not with the HIV exposure focus particularly, the boys and the girls seem to be, so the boys uniformly do worse than the girls across the group. And the boys and the girls seem to be more sensitive to slightly different risk factors. So I'm just struggling. I don't have to remember offhand, but I think some of the more physical risk factors, such as anemia, seem to affect the boys more than the girls, and for example, a birth weight tends to, we know, is well associated with developmental and cognitive outcomes. And those who are very low birth weight and those who are very high birth weight of the boys seem to be at particular risk of poor development. So these are quite complicated relationships, but there does definitely seem to be a difference between the boys and the girls, and it is something that we're paying attention to.
Thanks for those questions and for the insightful answers, Prof. Donald. We appreciate everyone who's come to this talk. It's really been a great collaboration between our two clubs, and hopefully the first of many more to come. However, this collaboration hasn't quite ended yet. We have a virtual pub session which will take place on Zoom in the next 15 minutes. So we'll start at 5:15 South African time and 4:15 UK time. In this virtual pub, we can ask more informal questions. It doesn't have to be directly related to the talks, but generally about what it's like to be someone in science and having any general advice about a career in neuroscience in general. So thank you everyone for listening. I've uploaded the link to the virtual pub chat in the chat room here. So please just feel free to connect on that link in the next 15 minutes. It'll give you a bit of a break and a breather to have a drink or so. Okay, thanks everyone.
Cup of tea.
Thank you. Thank you very much. Thank you.
Thank you.
Thank you.
Thank you.